I'm going to talk about resilience and PRRS, and in something that we put together which we call the Natural Disease Challenge Model, and I'll try and explain why we did that as I go along. It's actually part of a bigger project. It's our second Genome Canada project in swine health, and this is the title. It's led by Michael Dick, one of my colleagues at the U of A, John Harding, who many of you will know at the U of S at the Western College of Veterinary Medicine, and Bob Kemp on behalf of the PigGen Canada members. There are many elements, many components to this project, and some of them I've written there very quickly, from vaccine response, even some nutritional studies, how we can alter the gut microbiome by changing the nutrition and how that may then change immune response, organoid models, and more on the pregnant guilt challenge, which you heard a little bit about before, which was also part of the first Genome Canada project. And in that special edition of Vet Micro that Bob Rowland mentioned at the beginning, there is a review of what we learned from that PRRS model. Okay, so a natural challenge model for disease resilience in pigs, and again, many people involved. You can see some of them there, some of them you've heard speaking today. An international effort again, and at the bottom there in terms of the roll call are three of the students working on the project. Austin Putz is here in the audience. He works at Iowa State, and I'll show a little bit of his results, and then two of my students at the U of A. All three of them, I'm pleased to say, have spent significant amount of time in the barn where we do this work. I always like to see that they really get involved in, well, how is this data the primary data being generated? Okay, what is resilience? Probably you have some idea by now. Andrea Wilson called it an umbrella term. If we go to the dictionary, or a dictionary, the capacity to recover quickly from difficulties, toughness. And Bob Rowland pointed out some laggards, or losers as he tells me he calls them. But I was always fascinated I haven't got those pictures here, but you saw them several times earlier. I was fascinated by the pigs at the right-hand side of those histograms, because they were the winners, or the growers. So most of those pigs you saw in those pictures were infected with PRRS, but they were at the opposite end of growth, exactly what Bob Rowland was talking about in terms of when we see the disease, that's the impact it has, and whether it's loss of feed or whatever, a tremendous loss to the industry. And so our definition of disease resilience in pigs is that ability to respond to the pathogens in a way that minimizes the impact of disease. I often think, I don't know whether about you, but if I get flu, I'm one of those people who takes a lot of water and sleeps a lot, and then I'm up and running again. Whereas I have colleagues who'll be in bed for a week. I'm one of those growers. You can see that, right? So I somehow, very dangerous to say on stage, of course, is I feel like I can throw off that disease very quickly. And those are the pigs that we became very interested in. So this model, first of all, we wanted to establish something we called a natural challenge model. And as I say, I'll tell you the motivation for that in a moment. And again, coming back to the grow-finish pigs, so not reproductive PRRS, but grow-finish, we want to determine the genetic basis of disease resilience, but also, if we can, the holy grail, of course, is to develop predictors of resilience, and particularly in terms of, and I'll show some pyramid diagrams, but if we can predict in the genetic nucleus herds who is going to be able to be resilient when challenged with multiple pathogens, when they're still in their high health status. That's what we're interested in. And Joan Lunny showed you some biomarkers that seem to be different before infection, and those are the sort of biomarkers that we're interested in. So one of our motivations was, and we're still asking this question at this meeting, so the WUR SNP or WUR or GBP5, it explains a significant amount of variation in growth and viremia. But if you ask the breeding companies, not all of them are using it to make improvements. So that's kind of mystifying in a way. There are some really good reasons, and you should grab them and they'll explain to you what they are. But one of the things is they're always thinking, well, if we select for something for PRRS, what's it going to do for another disease? That's always one of the issues, and that's partly from work that was done years and years ago in chickens, where if we selected for certain things, then we saw greater susceptibility to other diseases. But another reason is this one, is PRRS is a syndrome, it's part of PRDC, etc., etc. And so the impact is not necessarily what we see when we do a challenge experiment. There are many other factors involved, and that was partly, and also you've heard about the follow-on studies from the PHGC, where they're doing vaccination and they're doing co-infections, but they're also doing field trials. So that was one of the first, if you like, the second wave of models was, well, what happens in the real world? And I think Bob Rowland used that expression, real world. And so a long time ago we talked about special weapons and tactics in terms of going and getting samples from outbreaks. And you can do it. There's some great work published. Steve Bishop, for example, who we've heard about today, who helped us design this model, did some fantastic work with outbreaks and showed some genetic elements of around those outbreaks. But they're very, very difficult to do. Your SWAT team has to be in its helicopter on the pad with the rotors turning, sometimes, to get the samples that you want. So what we decided to do was how could we create that real world, but in a model where we could actually do all of the things that we wanted to do. And the more we run this model, the more interesting it becomes and the more we want to do, I would say. So those of you who are feeling generous, get ready for the next proposal. Okay. So, I've been saying for a very long time that health is the area of biggest opportunity in animal genomics, not just pigs, but animal genomics, because there is genetic variation, but it's incredibly difficult to get at, because you have to really measure these phenotypes. So you have to set up these special systems to do it. And one of the reasons it's difficult is because where we do most of our measurement is at the top of the pyramid, where we keep everything healthy because we want to move genetic improvement all around the world without the disbenefit of taking disease with us. So production down here is not the same as here. So that gap is something that we're really, really interested in. Okay, and here's another version of that. And again, just reminding you, what we'd really like to do is to have measurements that we can do at the top of the pyramid that are going to predict performance at the bottom. So that's the, as I say, the real target of what we're doing. Okay, so the model. So I'm going to go through some diagrams, and here's a little bit of a description of the pig and sample flow and how we set it up. So the first thing we did, well, you can see the second bullet there. We've thrown away all the biosecurity rules. We're running a continuous flow. And when we started with a test station that has been used as a high health test station for many, many years, high-performing pigs going through that barn. So the first few groups of naive pigs that we introduced into the model, we brought in cedar pigs from 2 or 3 carefully selected commercial units with the types of disease we were interested in. So they came in, I can't remember the ratio actually, it was probably 10 to 1 or 10 to 2, something like that. So we brought those into a nursery and introduced the disease with the naive pigs. You're going to see we have a lot of batches. Each batch is from a single company and herd. We have something between 65 and 75 pigs come in to the model every 3 weeks. They're all the same genotype in that they're large white landrace barrows. In other words, the byproduct from the dam side of production of the pyramid, and they all coming from high health herds. And we have 7 sources, so there are 7 genetics companies involved. So they're weaned and then transported, and currently the way it's set up, they're transported into an acclimation unit where we collect a series of samples prior to challenge. Okay. So this is how we designed it. You can see 60 to 75 barrows coming in, 21 days of age. They're kept in that secure, high health nursery for about 3 weeks, where we collect some baseline samples, and I think they're on the next slide. Then we bring them into another nursery just up the road, and there they meet the pigs that came into that same nursery 3 weeks before. And they're nose to nose, they're in separate pens, but they're nose to nose with the pigs that are 3 weeks older. And so it goes on through to slaughter. Okay, can you just go back? Is that possible? Yeah. So just to, I haven't said, we'll have approximately 3,300 pigs altogether when we've finished. We're north of 2,000 already. And everybody has a DNA sample, just as in PHGC. And this time we're genotyping everybody with 660,000 SNPs. As I say, we are adding different features to the project as we go along. So we're using infrared thermography. You can see a group of pigs here in the nursery to get some information on whether we can use infrared thermography to see the onset of infection. In the finisher, we have, you'll see is very important, we have individual feed intake and also some trial equipment in terms of individual water intake. And sometimes we're using some enrichment experiments. In this case, we have accelerometers, but they're in the toys. So we're measuring the interaction of pigs with enrichment toys and whether that has any impact on what we're measuring. Okay, so you can see the raft of samples that we're collecting, 6 bleedings, as Bob Rowland said, and I think it was Lee Hood who I first heard talk about blood being a window on health, and I think it's relatively non-invasive, it's easy to collect, and we can divide that sample up into many different assays, and that's exactly what we're doing. Most of these are around immune response or measures of the cells in blood, so complete blood counts. Serology, you can see we're doing the fecal microbiome. We're good learners, Bob. We're really, that was one of the things that there was the philosophy of PHGC, is we're going to future-proof our sample collection, because what seems impossible in 2007 will be reality in a few years' time. And that's exactly what happened. You saw an example of that with the identification of the GBP5 mutation, which was done through transcriptomics. Okay, So hopefully you've got the flow. First blood sample at day 26, the third one 2 weeks after the challenge or after the pigs meet each other, and then we take a later sample another 4 weeks after that. Okay, so we're generating a lot of data, not only primary phenotypes, which you could see here, but also these secondary phenotypes, including the metabolome, the proteome, gut microbiome. So all of those things, and we're banking everything, as you heard about earlier. And our aims are phenotypic prediction of resilience, but primarily genotypic prediction of resilience. So what diseases did we introduce? And there's a long list here. We do vaccinate for PCV2, but hopefully some of your favorite pathogens are on this list. So we have 3 strains of PRRS in the barn. We have flu, although it's not always active, and then many other diseases which are in the barn and are expressed in terms of the clinical signs that we measure. So here's just some examples. These are some PMs that my student Jian Lim did when he was visiting the barn, and you can see our interpretation of what's going on in those lung lesions. Okay, so again, just as we saw in PHGC, you can see, and because these are pigs which we're euthanizing or are dying, you can see the average mortality is at 78 days, but you can see a large range of growth rate, the laggards and the growers, or the winners and losers. And here you can see we have quite a relatively high percentage of diarrhea or scours, and then the biggest cause of mortality is respiratory disease, as you might expect. The other thing we do is we measure the number of individual health treatments. This is a living beast, and it requires a lot of management. So we do medicate on a group basis at particular times to moderate what's happening in the barn, to keep the model going. Okay, so predicting which pigs are resilient. These are some of the immune response measures that we're using, and I'm going to talk about one of them in particular, which is the first one, complete blood count. But before I do that, how are we going to score that phenotype of resilience? And these are some of the ways we might do that. We've used, particularly in the work I'm going to show you, we use simply growth and treatment combined. But we have some, as you saw in the finisher, we have individual feed intake on every pig that gets through to the finisher. And Austin Putz is doing some really detailed analysis of these curves, and here are some individual feed curves. The red spots are where the animals were treated. So we have that, and there's Austin's presenting a poster later this evening on number 63, which will show much more on this. And he's also been looking at the heritability of some of these traits, and you can see that we have the expected heritabilities and good heritabilities that I think show that the model is going to deliver the sort of results we're looking for. So here's one of the traits that we might use as resilience, and I think it has a lot of potential. Austin's presenting some further elaborations of this in his poster. But essentially daily fluctuation in feed intake and scoring how much variation is going on. And particularly, of course, because we're interested in sickness, are events like this, where there are several days where the pig is decreasing its intake or is going off feed. And you can see here the mean squared error of feed intake has a heritability of 0.22. So again, particularly interesting, and with the expected genetic correlations with the traits that we're interested in. So everything is looking pretty positive in terms of identifying animals that are resilient in this model. So here, if I take that part, so we've established the model, we're seeing heritable variation in the traits we're interested in, and we're certainly developing different phenotypes that we can classify animals as being resilient or susceptible. Okay, so here's another picture, in this case from Jack Decker's, again reminding you of the number of pigs in the system, the phenotypes, and the potential biomarkers that we're interested in. And one of those is a commonly used technique in diagnostic laboratories, so that's taking serum and doing, whole blood and doing complete blood counts. You can see the sort of fractionation that happens. We can measure on, and remember we're doing this before the pigs are challenged, then 2 weeks after, and then another 4 weeks on. So we can measure the numbers of neutrophils, lymphocytes, etc. And again, in this case, my student hasn't been using the MSE of feed intake as resilience. He's been using a combination of growth and health score. So from severe clinical signs with wasting to imperfect health. And he's been classifying his animals on that basis. And here you can see just 200 animals, and again the range of growth rates that we're seeing in the model. Okay, so here are some initial results. And the 3 blood bleedings, so blood 1, 3, and 4, for those of you who are really paying attention. So in blood 1, and blood 1 is in that acclimation nursery, we're seeing differences in neutrophils and lymphocytes, or percentages of those, and the ratio between resistant and susceptible animals. Although there isn't a difference between those that die or are euthanized. And that may be for several reasons. But we are seeing an interesting difference in bleed 1 in terms of neutrophils and lymphocytes. And here, this is a subsample of the animals, which we've selected to do some qPCR of candidate genes, or genes that we may be expecting to see induced in some of these conditions. So you can see the resistant animals generally have higher neutrophils and lower lymphocytes in a very different ratio. So one hypothesis may well be that these resilient animals are armed already and are staying ahead. If you like, a naive hypothesis would be when the challenge comes, they're responding very quickly and appropriately to that challenge, whatever it may be, whether it's PRRS or an associated organism. And then the susceptible animals are slow to kick off and are then seeing persistent infection. That's what one interpretation of these results. So we're trying to work with that and see if we can build up data around that hypothesis. So the other thing we've done with CVCs, and again, yes, I have put a health warning on this slide. So we can take the results of the CVCs, and remember, we have 7 batches of animals coming from different companies, and then we repeat those cycles. And so what we've done here is take the animals from cycle 1 and built a model based on their complete blood counts. In this case, it's using all of the CVC data and combining that into a model, and we are ending up with a prediction accuracy, or a correlation, if you like, of about 69%. So Not fantastic in terms of predicting, but an interesting accuracy. It includes the dead animals as well. And if you look at that prediction, you can see, although it's not perfect by any means, but you can see that those resilient animals are growing 25 grams per day faster. On average, they have fewer treatments, and they certainly have lower mortality. So something that we're quite interested in. The beauty of the experiment is, of course, we're going to have more cycles and the opportunity to refine the model. Okay, so as I finish, I think one of the things that I'm really interested in is the blood transcriptome and its opportunity to predict resilience. And Joan Lunny showed you an example of genes that are differentially expressed before infection. Here I've taken 3 different models. One is another natural challenge, but it was done in a commercial barn, more like a field study that you've heard about. The virus is a PRRS challenge experiment. It's probably one that Kansas State did. And the bacterial challenge is Salmonella in this case. But you can see in the middle there's a common set of genes which before infection are associated with resilience or healthier animals. So I'm really looking forward to digging into some of those transcriptomic samples. So the initial CVC results, I think, show initial promise as a predictor, and in this case it's phenotypic resilience. So we're looking to move forward and integrate the 2 approaches and look at different predictors with some of the feed intake phenotypes that we're generating. We're going to be adding the genetic analysis, and now all of the animals that we have to date have 660,000 SNPs on them, so we'll be doing GWAS and moving forward. So next, complete the data collection, integrate the analyses, and then we're going to be exploring the blood transcriptome. We're particularly pleased Jack Deckers has a NIFA grant that's allowing us to look at about 1,000 samples in great detail, including the metabolome. And then the model, I think, allows us to explore infectivity and look at the impact of environmental enrichment, and also it provides many other opportunities if we can get further funding to keep it running.